All right, here's the notes video help number 2, and we're starting on slide 14. And you can see the definition is an allele. So as I was mentioning before, you don't have to, in your notebook for full credit, you don't have to write the full definition, but I would like you to write at least the term allele. So if in your notebook I see the word allele, and then I've seen you trying to answer these questions here in the box, you're good to go. You can make the choice to add it later if you want. If you want to write the full definition, of course that's fine here. But again, all you're required to do currently is to type or write down allele. Okay, and don't forget though, you can always go to Google Classroom to get this, so you can refer to it. Don't forget. All right, so what is allele? It's a version of a gene. So a good example is eye color. In this picture, we have eye color. There's not just one eye color, but genes do control eye color. And if there's more than one color, that must mean there's more than one version of a gene. Scientists came up with a word to represent different versions of a gene, and that word is allele. So to be clear, let's write down our thinking here. So add a little text box here.
What are you doing, Matt?
All right, so we have the eye color gene, and according to this picture, so for situation number 1, we have 2 versions. And another way of saying the versions is, instead of saying versions, we have 2 alleles. All right? Our job, if we're going to complete this task, I'll do one with you, and then so you know how to do 2 on your own, but we have to name each allele. So it says provide a name for each gene. Well, we did that, eye color gene. And then it says a descriptive name for its 2 alleles. So there are 2 versions of the gene, so there's 2 alleles. Allele 1, I'm going to call, oh, it's eye shape. It's not eye color. Sorry, it's eye shape. Let's correct this. And allele 1 will be almond shape allele. And allele 2, sorry, spelling them wrong here. Let's see. And allele 2 would be round shape allele. Right? And then what you do is you just decide which one you think is dominant. Do you think more people have round eyes or more almond-shaped eyes? For me, I'm going to, just based on our student population, it looks like allele 1 is probably dominant, and allele 2 equals recessive. The more common trait or allele isn't always the dominant one, but it often is. But there's always exceptions. But in this example, I'm just guessing. So for this last part where it says decide if it's dominant or recessive, so when you do number 2 and you're looking at lip shape, just pick which one you think is dominant, or you know, it's up to you. But I'm not going to count off wrong if you chose dominant versus recessive. It's up to you. It's your choice. But when you answer number 2, it should look something like this one. So I've done one for you. I've made that an example. I use this same pattern to create number 2. All right. And again, it's all about alleles and using this word to describe different versions of a gene. All right. Our next slide here is more involved. This is an SOU, or a statement of understanding task. And it's a little technical. What I'm going to do is go to the website, show you how to get there, and the link is active right here. It's a big URL, so I wouldn't want you to try to type this up, but if you can copy and paste it, go to the right site, that'd be fantastic. And we'll go there and we'll kind of look at it. But before we do, when we set these up, before we even look at the simulation, all we really have to do is read the problem. And the scientific problem is, how do gametes, which we learned were sex cells like sperm and egg, how do sperm or egg or gametes receive half of their alleles or chromosomes? So the normal cells of the body have two alleles for each gene or trait. It's just natural, just like in this example here. We have, so there's usually, let's see here, let's go back. So as you can see, this person in this example had FF. or they had AA. Everybody has two alleles or versions of a gene for each gene or trait. We do is we just have two of each. We got one from mom, one from dad. The question is, how does that happen? How do we get two of each version when our sperm only has one? So we're going to watch a video and try to figure out how do sperm or eggs only receive half the normal number of alleles, or just one for each trait. All right, your initial response should be right here. I can't write in a response for you, but how do you think it happens? So you would say, I think or hypothesize that half, or that sperm or egg gametes receive half the normal allele amount because, so how do you, and you would just say because, and you'd explain how you think that happens. How do you think it is that sperm or eggs are only receiving half? What do you think is going on for that to happen? So you just describe in your own words. You don't need to know technical words. You should be able to say some of the vocab that we've talked about before, like alleles or gametes or genes. These are terms we've talked about, but you don't need to know any other future technical words. But what I'm going to do is show you right now how we're going to gather data to decide if your hypothesis was supported or not. Right here it says what happened, your observations. If we do this in class together, you can get notes or observations from other people, and this is where you could do that. If you're doing this assignment on your own at home or using Google Classroom, you really just need to have 9 observations of your own, so you can just not worry about it. Now, there's 2 ways to do this. You can either print this at home, this slide. You could print slide 15 as it is. You can come and request that I print a copy for you, or you can just take a blank paper and follow the pattern. Scientific problem, write a hypothesis, get 10 observations, and then discuss your findings at the end, right? And compare your findings to your original hypothesis. It's up to you. But let's go to the page. So we're going to follow this URL. We're going to head right over here. And I Google Sumanis Inc. independent assortment. That's what I Google to get to the right spot. And then the first result is the correct one. You get right here. All right, and then you're going to click on animation. And to do this right, you're going to click narrated. And your job is to click narrated, but take the sound off. So I've now killed the sound for the simulation. The guy's talking like I'm talking, and probably too much. You don't need to hear it. All you need to do is visually watch what's happening in the screen. I want you to know that this is a single cell. This cell's job is to make sperm and eggs. So if it was a man, it's making sperm. If it's a lady, it's making an egg, right? We're going to follow the journey of these chromosomes, and notice the letters here. Remember, letters represent traits, genes, or versions of a gene. This would be a recessive gene, little s. It's a dominant s. So these 2 s's are 2 different versions or alleles of a particular trait, 2 different versions of a gene. You have dominant and recessive. So you got a recessive allele here, dominant version here. And then these chromosomes are carrying the Y allele. This isn't the Y chromosome. What this means is in this simulation, they're just symbolizing Y as a type of gene or an allele. This would be a dominant allele. This would be a recessive allele. And all you do is hit play. And you watch. You can also speed this process up. You can actually control how it behaves. You can just see. So this could be your first observation would just be describing what you see here. But you quickly see that each chromosome duplicates itself. And that means each allele is duplicated. You follow this pattern. And this whole time you should be writing observations. Your 9 total observations. All right, right here is where I need to explain. So this says alternative alignments. There's 2 different ways that the chromosomes could be organized in the cell. This is the first possibility, where S's are on this side, Y's are on this side. And another way that they can line up is S's here, or the chromosomes containing the S gene here, and the chromosomes containing the Y gene down here. Right now, you have to only decide if you want to look at alignment 2 or alignment 1. You make that decision and you don't follow both. So you're just going to look at alignment 1 and describe what you see, or you're just going to look at alignment 2 and describe what you see. What I need you to do in your statement of understanding is to explain right here, somewhere here, you need to show I viewed alignment 1, or I viewed alignment 2. It's one or the other. Don't look at both. That'll confuse you. You'll get bad results if you look at both together and describe both. Just look at one and describe what you see. All right, I'm, for this example, just going to watch alignment 1. I'm going to ignore alignment 2, just study alignment 1. So if I was going by alignment 1, just to give you an example, I would say, well, the cell divided into 2. Now there's 2 cells where there once was 1. Again, I'm ignoring alignment 2. I'm only looking at alignment 1. And the original cell divided in 2, and you can see there's only 2 here when there used to be 4. So the original cell had 4, and then it divided, and they got separated. You had these things come out and grab them. These are all things you can observe and discuss. Boom. So the cell takes one of the chromosomes that had the S and puts it into one, and the other chromosome that had the S goes in the other cell. And the same for the Y. When it split, one of the chromosomes with the Y gene or the Y trait went over here. One of the chromosomes with the Y trait went over here. So now you have 2 cells that have different versions or alleles. This top cell has the recessive S allele and the dominant Y allele, and this lower cell now has the dominant S allele and the recessive Y allele. So they're not the same anymore. They're not identical cells because they divided and separated chromosomes and the alleles differently. You should be writing observations. I'm going to hit play again. And you continue to go, and you describe how chromosomes move, but you come up with your own observations. You can see now that it's become 4 cells. So 4 cells, each one may or may not be different. These are all things that you can discuss and report on. And again, I'm only looking at alignment 1. Alignment 2, which is this side over, I'm ignoring. It's not important. There you go. Now, something that the simulation doesn't tell you is, at this point, each of these represents a sperm or egg. And you have to ask yourself, does each of these 4 sperm or eggs, do they have the same number of alleles, fewer or more? So you compare one of these final cells, like this one right here, compare it to the original. And this was the original cell. This original cell made 4 cells. And did the chromosome number change at all? Yes, no. Is it more, less? These are things you can discuss in your hypothesis. So when you take all the observations you collected here, all 9 of them, you then decide, my original hypothesis was, is it supported or not supported? This is because, and this area after this is where you're going to use your observations to explain and to defend why you think your hypothesis was supported, or use your observations up here to explain why you think it wasn't supported. But you have to give evidence for what you're claiming. And if you're claiming your hypothesis is supported, you better use your observations to help explain how it is. Or if it's not supported, you need to use your observations, the 9 you had above, to say why it wasn't supported. The more evidence or observations you use to support your ideas, the better the grade. And any observations you take down here, they should be helping you understand this original problem. So all your observations should be based on the fact that you're trying to understand how half the alleles end up in the sperm cells. All right, good luck.